So traditional microbiology pipeline, the classical microbiology approach was built on pure culture isolation. Collect a sample, grow microbes on plates, isolate colonies, then sequence and characterize the genome of the cultured organism. This pipeline was revolutionary at its time. For example, cost postulates, but it had major blind spots. Most microbes refuse to grow under lab conditions, and even those that do behave differently when removed from their native community context. Even those that do behave differently when removed from their native community context. The result, early microbiology gave us insights into a few model organisms, for example, E. coli, Bacillus subtilis, but left the vast majority of microbial life essentially invisible. Now, why traditional pipelines fall short? So, the first limitation that we're going to be talking about is non-culturability. More than 99% of microbes in nature cannot be cultured with standard lab methods. For example, Mycobacterium leprae, that is leprosy bacterium, has never been cultured outside a host. Second limitation is that the loss of ecological context. Even if a microbe can be cultured, removing it from its natural community strips away the ecological interactions, mutualism, competition, quorum, sensing, horizontal gene transfer that shapes its real world function. In short, microbes we could culture give a distorted and incomplete view of microbial life. Now, why do we need metagenomics? So, microbial communities function as complex, resilient, and adaptable ecosystems where emergent properties only arise when the full network of interactions is intact. Studying single organisms in isolation is like trying to understand the rainforest by growing one tree in a pot. You miss the interdependencies, the nutrient cycles, the checks and balances that make the system resilient, right? Metagenomics therefore bypasses culturing entirely, sequencing DNA directly from the environment, soil, ocean, gut, skin, etc., preserving the ecological snapshot. And what is a genome? So, a genome is the complete DNA content of a single organism, including all its chromosomes and plasmids. It encodes the full set of instructions that determine an organism's identity, metabolism, and adaptation. Genomes were the foundation of the genomics era, 1990s to 2000s, but they only describe individual species in isolation, not communities. They were the foundation of genomics era, but they only describe individual species in isolation, not communities. So we cannot see the interdependencies between these species. Now, what is a metagenome? So, a metagenome is the collective genomic content of all organisms in a community, sequenced together as a bulk sample. So, here we can see the interdependencies between the organisms of a community. A metagenome is the collective genomic content of all organisms in a community, sequenced together in as a bulk sample. Instead of telling us what one bacterium can do, a metagenome tells us what the community as a whole is capable of. For example, in soil metagenomes, uh, metagenomes, one species may encode enzymes for cellulose degradation, another for nitrogen fixation, another for phosphorus solubilization, together forming a functional ecosystem. Metagenomes therefore reveal not just diversity but functional potential of ecosystems. Now, what is a microbiome? So, the microbiome is the set of microorganisms in a given environment combined with the genomic content and functional capabilities. Crucially, it is more than a census of organisms, it captures the ecology plus the molecular toolkit. For example, the gut microbiome does not just mean the bacteria living in the intestine, but the sum of their collective metabolic abilities, fermentation, vitamin synthesis, SCFA production, and their ecological interactions with each other and the host. Now, what is a pangenome? So, a pangenome is the union of all genes found across all strains of a species. Union of all genes found across all strains of a species. It highlights that even within one species label, there can be enormous functional diversity. The core genome includes genes shared by all strains, housekeeping, essential survival. The accessory genome includes strain-specific genes, antibiotic resistance, virulence factors, niche adaptations. For example, Escherichia coli has a relatively small core genome, around 2000 genes, but a massive pangenome, around 18,000 genes, meaning two different strains of E. coli may share less than 50% of their genes. This underscores why strain level profiling is essential for precision medicine and surveillance. Now, moving on, the first question in metagenomics, who is there? The very first challenge in metagenomics was to identify the taxonomic composition of a community, which organisms are present and in what relative abundance. This is not trivial because sequencing produces millions of anonymous fragments that must be classified, and algorithms like Kraken, Metafine, and Kaiju were built to solve this. Answering who is there enables biodiversity surveys, ecological comparisons, and clinical diagnostics. However, it also opens up the problem of novel organisms. What if a sequence belongs to a species or even a whole lineage never seen before? New discoveries enabled by metagenomics. So, metagenomics has dramatically expanded our view of the tree of life by uncovering microbes we never knew existed. Examples include the candidate phyla radiation CPR, an entire group of ultra small symbiotic bacteria only revealed through metagenomic assembly, and the discovery of giant viruses like Mimi virus and Pandora virus. These discoveries reveal that Earth's microbial diversity is vastly larger than what culture-based studies ever suggested, and that our reference databases still capture only a small fraction of microbial reality.